Hello, good morning, good afternoon, good evening, wherever you are. Here we are in another session diving into the world of rejuvenating our DNA, the anti-aging technologies. This is the fifth video of the course, and today's topic is DNA amplification, Gigaprep. Well, in plain words, growing bacteria cells while they make lots of copies of DNA and afterwards extract the DNA. And Gigaprep is just the commercial name of the kit that help us to extract the DNA. Easy? Let's repeat the concept to fix it in our minds. It's just growing bacteria cells that has the desired DNA inside. At the same time that the number of bacteria grow, they replicate the DNA inside, the plasmid DNA. And afterwards, we extract the DNA using a commercial kit that is called Gigaprep. This is a very important step in the process. We need DNA of high quality to carry on with the following steps. Well, in order to gain some context, I would recommend you to see the first video. It was an overview of the science behind this technology, the steps necessary to produce EV with OSK. Remember, today I will remind you briefly the content of the first video. We need some context in case you have just landed here. Well, after the reminder, we will submerge again in this technology. In this video, the fifth one, we will learn to grow bacteria cells, as you see here, in suspension cultures. And while they grow, they replicate, they duplicate the DNA. We get lots of copies of DNA, lots of copies of the three DNAs. Remember, the DNA with OSK, the DNA with the RepCap genes, and the DNA with the helper genes. Okay? And after growing, replicating, or making a lot of copies, we will extract the DNA using a commercial kit that is called Gigaprep. That is today's topic. And here it is, Eric Amado, ready to guide you in this field, PhD, scientist representing Novosumer, a platform to share the science behind the anti-aging technologies, all that you need to understand how to stop and revert the age of the DNA. And most important, how to do it yourself. All that you need, material, equipment, steps will describe everything. Everything in novosumer.com or YouTube. In novosumer.com in the following months it will be released, okay? Okay, let's go for the today's topic. DNA amplification, Gigaprep. Let's go for the slides. Well, let's remember in a couple of minutes what we learned in the first video. We learned about the DNA, this beautiful molecule inside our cells. It contains information to shape our bodies, to build everything. Your cells read the DNA as a manual to build all their components. Okay, just remember, the DNA is just information. It's a guide to build things in our bodies. Okay, after this reminder, let me ask, do you remember something else? Maybe these little marks in the DNA? I show you that the DNA is modified with these little marks that you have here. Have a look. You see the little marks, right? The marks are stamps. They provide extra information. And sometimes they say, this DNA is old. Don't read it. It's a shame when you read it. Don't you think? When our cells see the marks, they literally read, this DNA is old. Don't read it. Well, when this happens, our cells lose functions. They get older. That is correct. As time passes, the amount of marks increase. Do you see here? This DNA contains lots of marks. As time passes, the DNA piles up a lot of these tiny marks. Well, in this DNA, the one with lots of marks, the information is no longer available, is no longer read, and our cells get older because of that, as a consequence of the marks in the DNA. That is correct. Our cells stop reading parts of our DNA. Well, it's when you get older. I'm sorry, my friend, but don't worry. Our cells have a solution for that. It's called OSK, a magic potion to revert the age of the DNA. OSK clean many of these marks. OSK polish the DNA. It reduces the amount of marks, and the cells are again able to read the DNA. In other words, we rejuvenate. Oh yeah, we rejuvenate thanks to OSK. Great, right? Good to know. Well, that is the key of this technology. We provide OSK to the cells to rejuvenate them. We reactivate somehow OSK and young again. OSK, young and healthy again. Cheers! Okay, let's remember how to produce OSK. Let's go for the next slide. Okay, this slide is a little reminder from previous videos. This slide is a summary of the steps necessary to produce EV with OSK inside. Well, before starting, we need nice DNA. We need to design good DNA. DNA with the OSK genes, DNA with the RepCap genes, remember, necessary to produce the EV particles, and helper genes to help all the process. So the first step was inserting the DNA inside the bacteria cells. You see here? That was the first step. It was called transformation. And the next step is growing bacteria producing millions of copies of DNA as they grow and extracting this DNA. And we extract the DNA with the Gigaprep kit. Okay? This is today's topic. Growing bacteria producing millions of copies of DNA and extracting the DNA. Well, this is the first step, in fact, in the process of producing OSK. Inserting DNA, producing copies, and extracting the DNA. And what is the second step? The second step is encapsulating the DNA in EV particles, and we do it inside a second type of cells, mammalian cells. So we insert the 3 DNAs because these 3 DNAs are necessary. We introduce the 3 DNAs inside mammalian cells. There they are, OSK, RepCap, and helper. So once inside, the 3 DNAs work in cooperation. They produce copies of OSK here, as you can see. Afterwards, RepCap produce EV particles. Here you are. And then the OSK genes are included, encapsulated in EV particles. And all the process is eased by the helper genes, okay? And easy peasy. Now we are in the third step. The third step is just cleaning the EV particles, is purifying the EV particles. Here you are. Easy peasy, right? Let's go for today's topic. Growing bacteria, producing millions of copies of DNA, and extracting the DNA with the Gigaprep kit. Well, we are finally in today's topic, DNA amplification and extraction by the Gigaprep kit. And these are the 4 steps that we are going to follow to do so. Let's have a look here. There are only 4 steps. Are you here with me in the first step? Okay, the first step is just picking one of the colonies from the transformation plate and transferring the colony to a flask. This flask is full of rich medium with antibiotic. So the bacteria cell with the DNA inside will find here a rich medium to grow. And while growing, it will amplify the DNA. The bacteria will duplicate once and again the DNA. Okay? But this is the second step. The second step, as I have mentioned, is grow the bacteria culture. The cells will grow and produce many copies of this DNA, and we provide the optimum conditions to grow. So as you saw here, we had the colony inside the flask, and we provide the rest of the conditions to grow, which are 37 Celsius degrees, 16 to 24 hours, and agitation. The chamber, do you remember the chamber? The incubation chamber? The incubation chamber will provide the physical conditions. The 37 Celsius degree, this steady temperature, and some agitation, some movement. And while growing, as you can see in number, the bacteria cells will produce many copies of the DNA. Understood? Easy? Okay, let's go for the third step. The third step is harvesting the culture. Okay, we need to separate at this time the cells from the medium because we need to recover the DNA that is inside the bacteria cells. So we use a centrifuge. The centrifuge will produce a circular movement, and this circular movement at high speed will help us to separate the cells that you see here from the medium. We will discard this medium and we will take the cells that you see here. Okay, so the first step is harvesting, is recovering the cells by using a centrifuge. Okay, let's go for the fourth and last step, using a Gigaprep kit to extract the DNA from the bacteria cells. Okay, the Gigaprep kit will help us to break the membrane of the cell. Afterwards, after breaking the membrane, all the content of the cell will be available. So this kit helps us to kidnap the DNA, to take specifically the DNA from the mix. And it will help us to clean the DNA from the rest of these contaminants. And after cleaning the DNA, we will use certain solutions to release the clean DNA. So at the end of the process, we will have tons, millions and millions of copies of DNA clean from contaminants and ready to work, ready for the following steps of the process. Easy? Let's go step by step to understand the whole process. Let's go for the next slide. Well, here we are in the first step, an easy and quick step. The number one, picking a colony and transferring the colony to a flask with LB medium or another rich medium in which the cell feel happy. So what do we need? We need the flame again to guarantee a sterile environment. We need the agar plates, the LB agar plates with the colonies, as you can see here, the colonies that grow. We need an inoculation loop is just a piece of plastic. You can use the tip of a pipette instead, or a metallic one. They look like this. These are cheap, nice, and also they are long enough to touch the LB medium. So what is this step? Is just picking one colony, which means just touching with the tip of the pipette or the tip of the inoculation tube, and transferring it to a flask with LB medium. The LB medium will be something like this. Just touch the surface and stir a little bit. It will be enough. Happy? Easy? Super quick and easy step. Let's go for the second step. Well, here we are in the step number 2, growing the bacteria culture. It means providing optimum conditions for the culture to grow. And as the bacteria grow in number, as you can see here, it amplifies the number of copies of DNA. So at the end of this step, we will have millions of copies of DNA. And what did we do? We just place the the flask inside an incubation chamber, and the incubation chamber will provide the rest of the conditions, which are 37 Celsius degrees, a steady temperature over this time. The time is 16 to 24 hours and 200 RPMs. You can see here in the picture the conditions as well. You see here 200 RPMs, the temperature, and the flask inside the chamber. The real chamber look like this. This is a real chamber. And one thing that I forgot is the antibiotic. Remember, in all these mediums that we are using, the medium that is here inside the flask has antibiotics. And remember, we need the antibiotic to select cells. We only want cells with DNA to grow. So only cells with the DNA that we want will grow. Okay? Easy step, right? Let's go for the third step. Step number 2. Number 3. Sorry. Let's go for it. Okay, the step number 3. Harvest the culture. Harvest the culture is just separating the medium from the cells. After growing the cells, we want to separate the medium that is no longer necessary from the cells. And a centrifuge will help us to separate the cells from the medium. As you can see here, once you place your bottle inside the centrifuge, it will spin, and the centrifuge force of the spin will help us to separate the supernatant, as you can see here, from the cells. Easy, right? Okay, what we need for this step is your culture. As you can see here, it's opaque, it's less transparent because the cells have grown very much. Okay, you need your culture, and you need an empty bottle, an empty centrifuge bottle, a special bottle for centrifuges. So we need both things, the culture and the bottle. And what we are going to do is pouring the culture inside the bottle, as you can see here, close to the flame to guarantee the sterile conditions. Okay, you have your bottle with the culture. We close the bottle. Here it is. And what do we do? Is placing the bottle inside a centrifuge. This is a picture of a centrifuge. It has a rotor here, and we will place the centrifuge bottle inside one of the holes. And this is a display. This is the display of the centrifuge. We will set up the machine through this display. And what are the conditions for the centrifuge? Are 7,000 Gs, 20 minutes at 4 Celsius degrees. Okay, so we will set up the centrifuge under these conditions. We will start it, and after 20 minutes, we will see as the supernatant is separated from the cells. Okay? After the centrifugation, your tube will look like something like this or this. These two figures that you see here, this is a real picture. You see here the cells? The cells are very compact. This is called a pellet of cells, and the supernatant is in the surface on top of the cells. So now we have what we want, that is the cells. So we discard the supernatant in a special bin, a bin for biological residues. So after discarding the supernatant, we will have our cells. And we have two options with these cells. We can freeze them or carry on with the next steps. Okay, in order to work with this pellet, we have to scrape it with a plastic tool, a steady plastic tool. And that's it. We take the cells, and end of the story, we can carry on to the next step. Okay, we are in the step number 4, the Gigaprep, extracting the DNA with this commercial kit. Well, there are other techniques that can be used, like sodium chloride gradient or quattro, or well, many other alternatives. But the Gigaprep is the one that we are using here. It's a kit that helps us to extract pure DNA, and it consists of several steps. Let's go. Let's go to see them briefly, okay? But we will explain them more in detail in following slides. The first slide, sorry, in the first step, which is called the cell lysis, what we do is breaking the cells. On breaking the cells, they will release the DNA, as you can see here, and other components that are kind of contamination because we don't want them. You see here in a real picture that the cells break down and they form kind of clumps. So in here you have remainings of the cells, and here you have whatever they release in the liquid. That will be the DNA and other components. In the following step, what we do is using um, a resin, kind of material that kidnaps the DNA. So here you can see this part of the kit is a resin, a special material that kidnaps the DNA, and sometimes a little bit of the contaminations. Here you are. As you can see, these are the liquid with the DNA and other contaminants, and this part here is the resin that captures the DNA. The liquid will pass through this device that is kind of a filter and will trap the DNA here, as you can see in the picture. You see here, right? Okay, so now we have in the kit the DNA and some contamination. So what we do is wash the DNA. We wash the resin. So we pass a liquid through here, and it will dissolve, it will dilute the contaminations, but the DNA will still remain here because the resin is very specific. It traps, it tends to kidnap only the DNA. This resin is positively charged and the DNA is negatively charged, so uh, they interact strongly. You see it here, okay? A real picture. The DNA will be trapped here in this section in the resin of the kit. Okay, so you have the DNA, only the DNA trapped in the resin. And now we want the resin, the DNA. So what we do is eluting the DNA. We freeze the DNA from the resin because we only want the DNA. So what we do is pour in liquid with high concentration of salt. And the high concentration of salt will help us to get the DNA freed from the resin. As you can see here, do you see? Yeah, it's just liquid with high concentration of salt and your DNA. Okay, now you have a solution and DNA, and you don't know the concentration of the DNA. So what we do is concentrating very much the DNA, and we do it by lowering the solubility of the DNA. We use substances to reduce the solubility, and it will precipitate. It will form kind of crystals, kind of lamps here, as you can see here. Once it's precipitated, we just discard the liquid and we will have the DNA like this, as you can see here, like kind of a clump, kind of a pellet of DNA. This will only contain DNA. So now that we have only DNA without liquid, now we can add as much liquid as we want to reach a specific concentration. So what we do is diluting the DNA in water or another buffer that guarantees the stability of the DNA. So we just add a little bit of water, another buffer, and we will get a concentration of DNA that we want, that we desire. Easy? Let's repeat it. We break the cells to release the DNA. We trap the DNA with a resin. The resin will have the DNA and some contaminant, some contamination. So we wash the resin to get rid of the contamination. Now we have pure DNA here and we want to recover the DNA. So we use uh, water with a high concentration of salt to release the DNA. So now we have this solution with a lot of DNA and a lot of salt. And we want to get rid of the water or of the liquid, and we precipitate it. We add a certain solution to precipitate it, to leave it as crystals, as a pellet of DNA. After having our pellet with the DNA, we will dissolve again the DNA in as much liquid as we want to reach the concentration that we want. Easy? Let's see in detail these steps. Okay, let's go for it. Okay, let's break this down. The We are in the cell lysis step. Okay, so do you remember we have the bottle here with the pellet of cells? Okay, so the first thing we have to do is scraping the cells and transferring the cells to another bottle, to a bottle suitable for this purpose. Okay, so we can use a spatula or a plastic knife, right? Here you are. In here we are using a spatula to scrape the cells and transferring it to another bottle. And well, the first thing that, well, one of the things that we do is we add resuspension buffer. It's a buffer in which the cells are comfortable, and this buffer is provided by the kit. It's called a resuspension buffer. Here we are. We are transferring with this pipette the resuspension buffer to the bottle. What do we do next is resuspending. For this purpose, you can steer the spatula a little bit, or you can gently invert the tube with the cells, but you have to do it gently, okay? Gently. As you can see here, you resuspend the cells. That means distributing very well the cells in the resuspension buffer, okay? So that's it, just resuspending. When you see that everything is homogeneous, that your suspension is homogeneous, you can carry on with the following step. Okay, let's read here. Addition of lysis buffer, 125 milliliters. So the only thing that we have to do here is adding a lysis buffer. Okay, the lysis buffer have components that break down the cells, proteins, RNA, and other components. Okay, so here you have a representation of what is going on in the cells. The cells are breaking and they are releasing the DNA and other components. Okay, that is your solution. This is your These are your cells, the suspension buffer and the lysis buffer. It starts forming kind of lamps, okay? Because the cell walls of the membrane and other components are getting together and they are releasing the DNA that will be around here in the liquid. It will be well diluted in the liquid, okay? And you will start seeing these lamps. This is okay. That is perfectly fine. So what we do after adding the lysis buffer, we incubate the resuspension with the lysis buffer for 5 minutes. It's important to respect this time, okay? Just 5 minutes. Just 5 minutes, remember. This will happen in 5 minutes. The cells will break and they will release the DNA. Yeah, I have told you, we just use 5 minutes because it's a strong buffer. So what we have to do is neutralizing this lysis buffer, make it less harmful for the DNA. Once the cells are broken, we neutralize the buffer. So we add neutralization buffer that will neutralize the solution. Okay? So now we have the buffer and the suspension neutralized. What do we do next? We transfer all that we have in the resuspension to one of the Gigaprep Kit cartridges. You see here the cartridge and a bottle. The bottle is attached to the cartridge in the bottom. And in here, you see the cartridge, right? It's just in this occasion, this cartridge is just for filtering. Okay? So we add this here to the cartridge that is going to be filtered. Well, now we can go for the next step. You see here is the filtration. But before filtration, we leave the suspension here for 2 minutes with the neutralization buffer. This will settle the resuspension. Okay? The resuspension will get used to the cartridge and the neutralization buffer will neutralize the resuspension effectively. And what do we do next? We just filter it. Okay? How do we filter? Do you see here the cartridge? The cartridge has here a little tube. So we have to couple a filtration pump. So once we attach the filtration pump, it will exert pressure and the liquid will go this direction. You see here? In here you can see the liquid very, very clear because it doesn't have the rest of the components. Let's say debris or other things that we don't want. Okay? So let's repeat this step. We just let the suspension here for 2 minutes with the neutralization buffer. We attach the pump. The pump will exert a pressure and the pressure will make the filtration possible. So the suspension will go through the cartridge to the bottle, and it will be kind of cleaner. It will be more clean. Okay, so that was all. Let's repeat everything so that you have it well in mind. So first thing, we transfer the pellet to another bottle. We add a resuspension buffer. We resuspend the cells so that they are homogeneously resuspended in the buffer. And afterwards, we add the lysis buffer. The lysis buffer will break the cells. This is the key step. It will break the cells and release the DNA and other components that are called contaminants. After the addition of the lysis, we add the neutralization buffer to neutralize the lysis buffer, and we transfer all the resuspension to a cartridge, the filtration cartridge. After the filtration cartridge, after transferring it to the filtration cartridge, it will stay there. We will incubate it for 2 minutes. And finally, we will filtrate the resuspension so that we have the DNA here and some little other contaminants in here. Okay, but it will be mainly the DNA well resuspended. Okay, let's go for the next step. Let's go for it. Now we are in the DNA binding step. It's just binding the DNA to a special resin that binds specifically the DNA, that kidnaps the DNA. This is basically this step. Let's go through it. Let's break it down. So remember, from the previous step, we have here the suspension filter. And we have here the filtration cartridge with the remainings of the cell, the things that we don't want. Here there is a picture, a real picture of this with the suspension here, with the DNA and some contaminants, and the remainings of the cell uh, over the over the filtration cartridge. So the first thing that we do is just getting rid of the filtration cartridge. We get rid of it. So we bin the filtration cartridge and we keep the resuspension with the DNA and, well, some contaminants. It's obvious that there is some contaminants. Here you have a real picture of the bottle with the suspension with the DNA. And what do we do next? We just pour the suspension with the DNA into a new cartridge. This cartridge is the DNA binding cartridge. So this is the cartridge that binds the DNA specifically. Do you see here the cartridge? Do you see this gray area? Do you see this gray area? This gray area is the one that specifically interacts with the DNA. That's it. This trap the DNA. This kidnaps the DNA. Okay? And, well, below the DNA binding cartridge, we have a bottle. Okay? This bottle will capture things that pass through the cartridge. So this step is just pouring the suspension with the DNA inside the DNA binding cartridge. Okay, so what do we do next? It's just incubating. Incubating means leaving the things like this for 5 to 10 minutes. Why? Because we want the DNA to get used to the resin. It will start permeating a little bit the resin. It will start interacting for 5 to 10 minutes. This is a real picture, okay, with the resuspension with the DNA and some contaminants incubating. So in here it will be for 5 to 10 minutes. And after the 5-10 minutes of incubation is just the filtration. For this purpose, we attach a filtration pump here to the cartridge, okay? Here you see, this is a little tube and we attach the filtration pump to this little tube. Okay, so after attaching the filtration pump to the cartridge, we will start the pump and it will start filtering the suspension. It will go through the DNA binding cartridge. Okay, what happens here? The process you see here, this is the resin, the DNA binding resin, and it has captured the majority of the DNA, but there is still some contaminants, okay? Little quantities of contaminants. It's okay. The majority of what we have here is DNA. So the majority of the things that have been attached to the resin are DNA. Okay, so at the end of this process, we will have in the flow through the majority of the contaminants, and we will have the majority of the DNA attached to the resin. That is what we want. Okay, so let's go over the steps quickly. So the first thing that we did is getting rid of the filtration cartridge. After getting rid of the filtration cartridge, remember we have the suspension with the DNA in the bottle. So we pour the content of the bottle, which is the suspension with the DNA, into another new cartridge. This new cartridge, that is the DNA binding cartridge. We incubated it for 5 to 10 minutes, and after the incubation, we just activated a pump that was attached to the cartridge so that the contaminants went through the cartridge and the DNA got bound to the resin. Okay, so at this step we have the DNA well bound to the resin and little contaminants. So let's go to the next step. In this step we are going to wash the DNA. Okay, just washing the resin with the DNA, so that we can get rid of the rest of the contaminants. You see here, this is the results of the previous step. We have the contaminants here in the bottle. So the first thing that we do is getting rid of the bottle. So we get rid of it, and we just have the DNA binding cartridge with the DNA attached and some contaminants. We attach a new bottle, another bottle, to the bottom, as you can see here. So now we have the resin with the DNA inside the cartridge, and we have a bottle attached to the cartridge. So what are we going to do now? We are going to use washing buffer, 275 milliliters of washing buffer. This is a buffer that has some salts, up to 800 millimolar of salt, sodium chloride, and is slightly acidic. It has a pH that is equal to 5.0. Okay, so what we are going to do now is pouring some of this buffer to the cartridge with the DNA. Okay, so we pour the washing buffer. As you can see here, whitish. Do you see it whitish over the resin with the DNA? Okay, and afterwards we incubate the whole thing. So the resin with the DNA and the buffer that we have just poured. Okay, we want the whole system to get used to the washing buffer. So we leave it for 2 to 5 minutes. Okay, it starts permeating the resin, and it starts interacting with the contaminants. Okay, so the contaminants start feeling comfortable in the washing buffer. And the last step is attaching again the filtration pump and filtrate. We attach the filtration pump to the cartridge, we activate the pump, and the liquid will pass through the DNA binding cartridge. You see here, this is the flow through of the process. the whitish one that is at the bottom of the bottle. As you can see here, now the flow through has the remaining contaminants. You see here? And the resin with the DNA is completely clean. So the only thing that we have now in the resin is DNA, pure clean DNA here in the gray zone, in the gray area. Okay, so the DNA is bound to the resin, very clean, and the remaining contaminants are in the flow through with the washing buffer. Okay, these last three steps have to be repeated. Okay, in order to be sure that is the DNA is super clean, we repeat these three steps. So we add again the washing buffer to the cartridge with the DNA. This time is very clean. We incubate it for 2 to 5 minutes because we want the contaminants. Well, there is no contaminants, but in case there is, we want the contaminants to feel comfortable in the washing buffer. And after incubating, we filtrate again. And this time is for real the last one in the cleaning process. Now we have the DNA super extra mega clean attached to the resin. As you see here, these small rings that represent the DNA are well attached to the resin and there isn't anything else. Okay? Okay. So I hope you are happy because we are going to go for the next step. Let's go for it. Here we are. Once we have the clean DNA, as you can see here in the previous slide, we have here the contaminants, right? So the only thing that we have to do is getting rid of this bottle with the contaminants. So we get rid of it and we attach a new bottle, that will collect the DNA, the pure DNA afterwards. So what do we do next? We add elution buffer, 100 milliliters. This buffer has pH 8.5, so it's slightly basic. 1.25 1.25 molar of sodium chloride. Okay, that will help us to elute the DNA afterwards. You see here. Okay, so we have a new bottle and the buffer here and the cartridge here. Okay, so what we do after adding the buffer is just incubating the cartridge with the buffer for two minutes. The DNA will get used to the buffer. Yeah, the buffer will start permeating the cartridge, the resin. Okay. So after leaving for 2 minutes, the buffer inside the cartridge, we will attach the pump here and we will activate the pump, but we will let only 30 to 40 milliliters to pass through the cartridge. Okay, once we see here in the bottom 30 to 40 milliliters, we stop the pump. We will start having here pure DNA. Okay, we stop the pump and we incubate again 2 minutes. We want to maximize this process. That's why we stop it and we incubate again for 2 minutes. And after incubating 2 minutes, we activate again the pump until all buffer pass through the cartridge. Allegedly, all the DNA, all the pure DNA should be here. But just in case, what we do to recover as much DNA as possible is repeating this process again. So we do these three steps 2 times. Okay, so we add again more dilution buffer, we incubated it for 2 minutes, we activate the pump, and we recover 30 to 40 milliliters. Again, afterwards, we incubate for 2 minutes, the remaining buffer, and we activate the pump. And after the second time, we can say that majority, if not all the DNA, all the pure DNA is here in this bottle, all the pure DNA. Okay, easy peasy, right? Let's go for the next step. Okay, so now we have 100 milliliters. Well, we have 100 milliliters or 200 milliliters, depending on whether you did it twice or not, the previous step. But the thing is, now we have the DNA here, the pure DNA. So what we have to do is discarding the cartridge. We don't need the cartridge anymore. Okay? And well, we have the bottle here. It will have 100 milliliters, let's say. So the next step is precipitating the DNA. That's it, making it less soluble. It will, we will make it to be in a solid state. Okay, to carry on with the following steps. So the first thing that we do is distributing the 100 milliliters in eight tubes like this. They are called Falcon. You can see here the Falcon tubes. So we transfer 30 milliliters of the DNA to each Falcon. So each Falcon will have 30 milliliters. And what do we do to insolubilize the DNA, to precipitate the DNA? Okay, we use isopropanol. So we transfer 30 milliliters of isopropanol to each tube. Uh, Please read here with me. The DNA is insoluble in isopropanol. It precipitates as a solid matter and goes to the bottom. So as we start adding the isopropanol, you will see as the DNA precipitates in the bottom of the Falcon tube. Mhm. But we want to gather as much as possible. We want to maximize this process. So what we do is centrifuging. Okay, so we put the Falcon tubes inside a centrifuge. We centrifuge it for 30 minutes at 4 Celsius degrees, and we set up the machine, the centrifuge at 12,000 Gs. Right? Understood? So the ultra centrifuge will be set up at 12,000 Gs for 30 minutes at 4 Celsius degrees. After the process, you will see this. This is the DNA. The DNA will get attached to the bottom as a pellet of DNA. So the only thing that we have to do is discarding the liquid, the isopropanol that is on top of this. We just drop it. Don't worry, the DNA will get attached to the bottom of the tube, so you will not lose any DNA. So okay, we have discarded the liquid, and now we have the DNA in the bottom of the bottle. What do? We uh, we want to maximize again the process, so we add 20 milliliters of ethanol at least 70 to 80%. And yeah, with this process, what we are going to do is cleaning. So we add the ethanol and we resuspend it properly. We shake it. We shake the tube gently in order to distribute again the DNA all over the bottle, all over the suspension, as you can see here. Okay. So let's repeat what we have just done. We have here the DNA, we discard the cartridge, we distribute the 100 milliliters or 200 milliliters in eight Falcon tubes. Once we have the Falcon tubes with the DNA, we add isopropanol. The isopropanol will insolubilize the DNA. So the DNA will go to the bottom. In order to maximize it, we centrifuge it, and after centrifuge it, we will see that the DNA comes to the bottom. We discard the liquid, all the liquid that is in the falcon, and afterwards we wash the DNA with ethanol. Is just adding 20 milliliters of ethanol, 70 to 80% of ethanol, and we shake it gently, or we can invert the falcon several times so that the DNA gets distributed among the suspension. It will be well distributed all around the suspension. Okay, easy. Let's go for the next slide. Okay, here we are at the very end of the process, solubilizing the DNA. So, okay, after adding the ethanol and resuspending the DNA well in the suspension, what we do is, because at this stage the DNA is insoluble, is in ethanol is insoluble. So in order to work nice with it, we centrifuge. So we again put the falcon tubes inside a centrifuge, and this time we set the machine for 10 minutes at 4 Celsius degree at 12,000 Gs. Okay, so we spin the suspension. And after spinning the suspension, what you will see in the bottom again is the DNA, very, very clean, washed with the ethanol. Yeah. And on top of the DNA, you see here the DNA, the pellet of DNA. On top of the DNA, you will have the ethanol. So what we do is discarding the liquid, discarding the ethanol. So you just can tilt the falcon and discard the ethanol. No worries. The DNA is well attached to the bottom, so you will not lose DNA. Okay. So you open the tube, invert it to discard the liquid. No worries, the DNA will remain in the bottom. I have just read it. Okay. So once you get rid of the ethanol, you use a tissue, a dry tissue. So you invert the tube over a dry tissue. Okay. And with this step, what we are doing is drying the DNA. So we dry the DNA, leaving it over the tissue for 30 to 60 minutes. No worries, the DNA will be still here, okay, at the bottom of the tube. You can see here the tubes, the falcon, drying over the tissue, dry tissue. Well, so now at the end of this step, we will have the DNA will attach to the bottom, it will be dry. So the next step is solubilizing the DNA. And what do we do for solubilizing? Is adding a TBE buffer, as you can see here, just 1 milliliter of TBE buffer. It will solubilize the DNA. So it will be soluble and not solid anymore. So we transfer 1 milliliter with the pipette, okay? And the next step is resuspending the DNA. Resuspending the DNA with the pipette. So we use the pipette, we just pipette up and down, up and down, up and down, up and down. And this process will help. At the beginning, it will be a little bit viscous, but as you pipette up and down, you will see that the suspension, the solution will be less viscous. It will be because it will be completely soluble. Okay. So we will have all the DNA very concentrated in 1 milliliter of TBE. We measure the concentration of DNA. We will use a nanodrop or whatever machine you have to measure the concentration of the DNA. A nanodrop is a good way, an accurate way to measure the DNA. So once we have the DNA measured, we will know how much DNA we have solubilized in the 1 milliliter of TBE. So what we do, and we want the DNA at a specific concentration for the following test steps. And the concentration is 1 mg milligram per milliliter of DNA. So I repeat, we want for further steps the concentration of DNA to be 1 milliliter. So what we do is just calculating the amount of TBE that we have to add in order to reach 1 milliliter. So we calculate the amount of TBE that we need and we add the TBE. Okay, we add the TBE. We resuspend it again very well with the pipette by pipetting down, up, down, up, down, up, down, up, down, up, down until we think that is well solubilized, and we measure the DNA. It will be close to 1 milliliter. 0.95 is okay. 1.05 is okay. So just try not to deviate more than 5%, okay? So at this stage, we will have a solution of DNA 1 mg per milliliter. Allegedly, we will have 8 bottles of this, so the only thing that we have to do is mixing the bottles or keeping them aside. Depends on the purpose or what you are going to do next. And end of the story. That's it, that's all. At this stage, we will have nice, pure, clean DNA at 1 milliliter, exactly what we need to carry on with the following steps. Okay, so that's all. Well, before finishing, let's summary all the steps all together. Okay, let's remember what we have just done in this video. Very last slide with a short summary of what we have done in this video. Okay, let's go for it. We pick one of the colonies from the transformation plate, as you can see. So we pick one of the colonies and we transfer to medium. We use an inoculating loop to transfer the colony to a flask with rich medium and antibiotics. Important. Okay. In the second step, what we did in the second step, grow the bacteria culture. The cells produce DNA copies while growing. They will copy many times the DNA. Okay, so for this purpose, we provide optimum conditions to grow. You remember 37 Celsius degrees, 16 to 24 hours, and 200 RPMs. That it is the shaking force. And as this process happens, the bacteria start multiplying, start growing in number. And as they grow in number, they copy many times the DNA. So at the end of this process, we will have a lot of cells with a lot of DNA. The next step, the third step, is harvesting the culture. It's just centrifuging to separate the cells from the medium. So we use a centrifuge to help us to separate the cells from the medium. As you can see here, we spin, we centrifuge the culture. Okay? And the fourth step, what is the fourth step? You know, it is just using a Gigaprep kit to extract the DNA from the bacteria cells. And this is the end of the process. This is the product of the process. A lot of DNA, pure DNA. Remember, a solution of 1 milligram per milliliter. Yeah, the Gigaprep kit is what we use to break the cells, kidnap the DNA, clean the DNA, and release the clean DNA. Easy? Easy? Okay, no, Sumer wants to go. I don't know where. He's saying thank you. Yeah, thank you. But before saying bye-bye, I have to remind you. I have to remind that you can find a more well-detailed protocol in novosumer.com. You will find it in the following months because it's not yet available the website. And it will be called something like Plasmid Amplification Step 2 Gigaprep. This protocol will be compliant with CGMP ISO 9001, ISO 45001, ISO 14001. So these protocols, or the protocols that you will find in the website, will be compliant with these standards for environment, for quality, and for health and safety. Okay? And now, yeah, it's time to say bye bye. Thank you very, very much for your time. Thank you very, very much for your attention. Just enjoy. Yeah, I hope you are an entrepreneur and want to implement these protocols. Okay? Thank you very much, my friend. Have a good day.